Hi everyone, my name is Vivian Xu, and today I'd like to talk to you about one project that I've been working on in the past year at Parsons New School for Design in New York City and also at GenSpace in Brooklyn. So the project's name is called Living Devices, and it was my master's thesis. So for time being, I was interested in the relationship between technology and life, and this question has although a lot of the technologies that we use right now to discuss this issue is sort of current and new, but this question has plagued us for centuries since Jacques de Vacus first made his mechanical defecating duck. So what I was interested was the border between the artificial and the natural, in this case, technology or artifice, and what we perceive as biological, and that is summed up by the concept of cyborg. This work that you see here is a very famous artist Stelarc, and it's his conception of the cyborg. But what I'm actually really interested in is interfacing organisms that are much more different from humans, and with specifically in my case electronics, because I think that they present a different aspect of life that is beyond a homocentric view of what that is and beyond our own comprehension. And I think that that life that is unknowable is very interesting and intriguing. So the main method that I was interested in was hacking into the living system through electricity, because since Luigi Galvani discovered animal electricity, it has been increasing proof that life itself is an electric system. I started designing these modified petri dishes where you can, the purpose of these would be you can culture bacteria, but at the same time plug in electronics that I designed that provided electronic stimulation, and you can control the voltage difference. You can create non-unified fields and dynamic fields to create an electronic environment for testing bacterial growth and seeing how that affects the organism's colonies. So this was a very early prototype. There were some issues that I ran into, like for instance, how you can, because electricity circuits need to be kept dry so they don't short circuit, but most wet media has a lot of liquid in it, and so I had to figure out a way where first the bacteria results would be preservable, and second, it wouldn't hurt the circuits or break them or cause any dysfunction. This is some of the experiments I did at GenSpace, which is a community biolab in Brooklyn, as Marianne mentioned before. So these three pieces are simple electronic circuits that I made. The one in the middle provides a single voltage stimulation, and the one that's on the far right provides two different voltage level stimulations. And so what it does is it creates a step of three for voltage, where the current can arbitrarily travel between these 3 points and balance them out. And so these are some of the results of my tests. As you can see, the agar, the chemical reactions happened within the agar that visualizes the electronic fields. And this specific experiment was very interesting because if you can see the top and the bottom, there's two dots. Those are bacteria colonies. But in fact, when I began the experiment, I seated the bacteria in the center of the plate, and they were pushed to the sides in a symmetrical manner by the electric fields. So I worked mostly with E. coli bacteria because it was one of the most common strains and accessible strains in the a level 1 biolab. But I'm also interested in working later on with bacteria, other types of bacteria that are more sensitive to electromagnetic fields. And then this also is a diagram of culturing bacteria within ecosystems. That's also something I'm interested in testing with. And also slime mold, because it's also a single cell organism that lives in colonies, but has a very different way of moving and communicating and growing. Thank you.